Hi everyone, and welcome to Lecture 1. In today's lecture, we cover a few different topics. We cover the review of some microbiology, basically the microbial world. What does it mean? We also then go into the chapter covering microbial anatomy, so some cell structure and function information. And then lastly, we cover metabolism. Now, to make the lecture a little easier for you to follow, or so as not to feel too overwhelmed, I'm going to break the video up into 3 smaller parts. Each part or each video will cover one of those 3 topics that we just mentioned. Now, when you go through these lessons, I recommend a few different things. One, it helps a lot of students to have printed copies of the slides where they then write their notes directly on the slide, so that they associate the notes that they took down with that exact slide and pictures and words. Another thing I recommend is really pay attention for clues. So for instance, when you look at the slides, you'll notice that certain terms are bold, colorful. You're going to want to know the definitions of those terms. I put terms on slides without their definitions so that you have to actually show up to the class, listen to the narrated lectures. You'll also notice that a lot of the slides have questions written on them. So throughout the notes, either as the title or as bullet points, I throw in some questions, but I don't write the answer. Instead, I give you the answer during the narrated lesson. So I highly recommend you write those questions down, especially in your study guides, for instance. Write the question down and then write the answer that was given to you. I also recommend that sometimes you'll notice I will slow down and I will repeat something a few times, okay? The reason I do that is to kind of help you realize, hey, boom, this is important. Let me write that down. And also because repetition will help you remember certain things better, okay? Now to start, what exactly is microbiology? So the first thing you want to ask yourself in this class is, what exactly is microbiology? So think about it for a second. How would you define microbiology? Okay. Think about breaking the word down. Micro and biology. So when you see biology, you know that that's the study of living things. And when you hear the word micro with something, what does that then tell you? Well, they're really small, right? So in this class, we are going to study living things that are very small, organisms that are not visible to the naked eye. So I want you to think about which organisms would that be? Well, usually there are 3 that we focus on: bacteria, fungi, and viruses. Now you may want to ask yourself, why would we want to study bacteria, fungi, viruses, any of that sort of thing? What's the benefit of studying them? Well, think about it. There are a few different benefits. First off is the obvious one, especially if any of you plan on going into nursing or the medical field. Well, the first thing that we can learn by studying these things is how to understand them and their processes so that we can determine how to destroy them or how to hinder them when they are causing problems to humans. Meaning, how do we treat humans who are being infected by the pathogenic ones of these microbes? Okay, so that's a big reason for studying microbiology, is the medical aspect, the understanding of these microbes to better treat patients. Another reason to study microbiology is the drug or antibiotic development aspect. Okay, so by studying these pathogens or potential pathogens, we're able to develop different drugs or antibiotics to help treat them. My personal favorite reason for studying microbiology is food production. So in the next slide, we'll kind of mention this a little bit too. Think about how many foods and drink products you wouldn't have if we never studied microbes or their processes such as fermentation, right? Think about things like yogurt, cheeses, breads, beers, alcohol, any of that, we wouldn't have without understanding microbiology. Lastly, microbiology is important for industry aspects. Okay, so the research field and obtaining different enzymes and various reagents. And we'll kind of touch upon these things throughout the course. Now, as I was just hinting to a minute ago, not all microbes are actually pathogenic or a problem. In fact, less than 5% of the microbes in the world are actually pathogenic. Okay? Most microbes either have no effect on humans or can actually benefit humans. So before we spend the course looking at various pathogenic or bad guy microbes, I want you to take a minute to kind of think about what are some beneficial microbes? Okay, how are some microbes actually helpful to humans? Now, we touched upon some of these a minute ago, for instance, food-based microbes. So being able to have things like yogurt, probiotics, right? In addition to that, one of the things we're going to keep mentioning throughout this course is your normal flora. There are tons of microbes covering the outside and inside of your body that actually help you every day. So one of the most famous categories of normal flora are your gut normal flora, right? If you've ever heard of all of those bacteria living in your digestive tract, well, you wouldn't be able to digest certain foods, including a lot of vegetable-based foods with cellulose in them, if you didn't have those bacteria, those microbes, using their enzymes to help you digest your food. Normal flora is also very important because think about it. If all of these bacteria are coating your body, then they're preventing the bad guys from being able to have access. So to continue on that thought, let's go to the next slide now. The idea of good versus bad microbes. So like I just mentioned, we all have normal flora, and I want you to circle this word on your slides because this is very important. It's going to keep coming back to you. Normal flora are all of the microbes on or in your body that are not associated with disease. Okay, not associated with disease at that moment. Okay, these are the microbes such as your gut normal flora that we were just talking about that are constantly living on or in your body and can actually help you, okay, by preventing other microbes from being able to live on your body. Now, the one downside about normal flora is that they can be what we call opportunistic pathogens. Okay, opportunistic pathogens, underline this word. Okay, this will keep coming back in this course. Opportunistic pathogens mean that even though normal flora are normally not bad guys, they are normally not pathogenic, under certain circumstances they can become pathogens. Okay, I want you to think about what kind of circumstances might make these good guys that are living on your body, just chilling on your skin or in your gut, what might suddenly make them a problem for you and give you symptoms? Okay, well, there are two main times when that will happen, when normal flora become opportunistic pathogens. The first one is if you ever become immunocompromised, right? So you're in a weakened state and these guys are opportunistic. So what are they going to do? They're going to jump on that chance, right? They're going to take advantage of your weakened state and now they will be pathogenic and hurt you. The other time that normal flora become opportunistic pathogens is if they end up in the wrong place. So for instance, if those gut normal flora, like E. coli, that we were just mentioning a minute ago, if they suddenly end up somewhere other than your gut, for instance, your urinary tract, well, boom, now you have an infection. You get a urinary tract infection because these microbes are not supposed to be there normally, and so now they are considered pathogens. Okay, so whenever you see opportunistic pathogens, remember that normal flora can become opportunistic pathogens if you are 1, immunocompromised, 2, if they end up in the wrong place. So the way that I usually like to kind of give the analogy for students to remember this concept is, well, you may be the nicest, best, genuinely good student in the world, right? Normally not a thief or a cheater. But if, let's say, you were sitting in the professor's office, they happen to have the final exam sitting in your line of sight, and well, suddenly some other student came along, right? Emergency came up, the professor had to quickly leave the room and forgot that that final exam was right in front of you. You're not normally a cheater, right? But I'm pretty sure that if you were suddenly left alone for a few seconds with the final exam just right there in front of you, there's a good chance that your cell phone's going to come out and you're going to snap a few quick pictures, right? Even if you're the most honest person in the world. And it's understandable because it's opportunistic, right? The opportunity came up and the temptation arose, okay? And that's what happens to the normal flora as well. Now, once normal flora or any microbe causes disease, that's when we call it a pathogen, okay? And there are certain terms associated with this idea of microbes causing disease. The first one is pathogenicity, okay? Pathogen being a harmful microbe, right? Pathogenicity then means the potential or the ability for a microbe to now cause disease, to be harmful, okay? So pathogenicity, what I would do if I were you, I'd underline that P, that first P letter in pathogenicity, and then underline the word potential. So pathogenicity is the potential to cause disease. Whereas virulence is after that. Virulence is the degree of pathogenicity. How severe of damage can that microbe cause? Okay, so pathogenicity is the potential to cause disease, whereas virulence is how severe of a disease will be caused. Okay, now the biggest culprits of diseases in humans tend to be nosocomial infections. Anyone who's already kind of experienced any clinical work probably have heard this term. Nosocomial infection simply means any infection picked up in healthcare facilities, such as in the hospital. Now, unfortunately, one of the themes that we're going to keep seeing in microbiology is that over 250,000 Americans a year die from sepsis, okay? Meaning that over 250,000 Americans each year die because they picked up an infection in the hospital, and sometimes that's going to be from those of you who are working in the medical field. Okay, so throughout this course, really think about how the stuff that we learn can help protect you, help protect future patients if you're going into the medical field, or even just help protect your loved ones if you're not going into the medical field, because everyone knows that at some point or another, you or your loved ones will have to walk into a hospital or a clinic or doctor's office, okay? So keep these things in mind that we go throughout the course. Now, if one of these microbes does become a bad guy, become pathogenic, it must be able to do 5 steps. And again, this is something that you'll see come back throughout the course. So make sure the next slide that we turn to, you circle, star, highlight. Okay, so like I just said, put stars all over this slide. It will keep coming back. Okay, when we think of pathogenic microbes, in order to be a pathogen or a dangerous bad guy, a microbe must be able to do these 5 things. The first thing they have to be able to do is enter your body or your cells. Okay, and think about it. What are some ways that they might enter your body? Well, breathing, right? You have to breathe all day. So any of those openings you're using to breathe, that's easy access. What else do you do all day? Eat and drink, right? Eating or drinking are easy access. Any cut that you get or opened wound, those are easy access. Okay? Sexual contact, that's another common entry for pathogens. Okay, so throughout the course, we're going to go through some of the various ways that microbes enter. But once they enter, in order to make you sick, they have to be able to stay in. If you instantly sneeze or, cough them out, well, then they wouldn't be a problem, right? They have to be able to attach and adhere, stay in your body. Okay, so now they've entered, they stay in. The next thing they have to do is defeat your defenses. Okay, so highlighting here the word defenses. What does defenses mean? In your body, that's your immune system, right? They need to be able to defeat your immune system, which is trying to destroy them. Once they've done that, they have to cause some sort of damage. Okay, because if they're not causing damage or not causing problems for you, then they're not a pathogen, right? You wouldn't even notice them. The last thing they have to do to be a true pathogen is transmit to a new host. Okay, because think about it. If this little bacteria only made one person ever in all of history sick, who never transmitted to any other hosts, well, then we wouldn't really know about it, and we wouldn't really consider it a pathogen or a problem. Okay? So just remember, in order to be a pathogen, a microbe must enter, stay in, defeat your defenses, cause damage, and then transmit to a new host. Now, in order for us to really understand how exactly microbes are able to be good or how exactly they're able to be bad, it's really important to know about their anatomy, basically the different structures and functions that they have that can then make them either really helpful or really problematic. As I mentioned before, we're now going to end this part of the video, and the next section of Lecture 1 is posted as a separate video. That way you have a chance to take a little break. If at any point you have any questions, you need any slide or information gone over a little bit extra, or you just want some practice problems for exams anytime you're going through lectures, feel free to contact me in the Remind app, day or night, 24/7. Like I always tell my students, I have no life. So feel free to contact me with any questions that you need a little extra help with.